Biuret test is not given by. First of all, what type of compounds show Biuret test? Remember students that Biuret test is given by proteins, right? So your Biuret test is the test for proteins. Do you know what is this test? It is that your protein is warmed with 10% solution of sodium hydroxide, that is NaOH, right? This is gently warmed, right? And to this a drop of copper sulphate solution is added. Right? Now there is a formation of reddish violet color. There is a reddish violet color appears. Now this appearance of reddish violet color indicates the presence of amide linkage or peptide linkage, right? Peptide linkages CONH, right? So wherever the CONH linkage is present, it gives Biuret test. Now in case of polypeptides, between the two amino acids, what is the type of linkage present? It is a peptide linkage, CONH, right? In urea also there is a peptide linkage. Say your urea, after heating urea, what is obtained is NH2 CONH CONH2, and proteins, the peptide linkages of course present, right? So wherever this peptide linkage is present, it will give positive Biuret test. So what is the thing which will not give Biuret test? Carbohydrates. Do you know the other tests given by proteins? It gives xanthoprotic test, right? The other test, the answer to this question is carbohydrates. And just for your knowledge, I'll tell you knowledge, or I would like to just refresh this thing, that the other tests given by proteins is, one is xanthoprotic test, what is done in this? It is your protein is treated with concentrated nitric acid, right? Now due to which they give a yellow color. On treating proteins with concentrated nitric acid, yellow color is obtained. So this is protein with concentrated nitric acid. What is obtained? Yellow color, right? So that is the same color is obtained when when by a mistake this nitric acid falls on the skin. This also gives Milan reaction, that is second reaction, or the third test which the protein gives is Milan reaction. What is it? This is Milan's reagent. That is, what is Milan's reagent? Students, this is essential for you to know. That's why I am telling you. This Milan's reagent is solution of mercuric and mercurous nitrates in nitric acid containing concentrated little nitrous acid. Right? So this is what is Millen's reagent. Fine. Now, if your protein is treated with Millen's reagent and heated, the solution gives red PPT, right, or red color. That is also a protein. Now we have got another test for protein, that is ninhydrin test. Now what is this ninhydrin test? Here the proteins or peptides with alpha amino acid give blue color with ninhydrin. Right? So here proteins or peptides containing and I can say alpha amino acids. Alpha amino acids means alpha amino acids give blue color with ninhydrin. Right? Your proline gives a different color with ninhydrin. So these are all the tests which are given by protein, and you should basically know these specific tests. Say here I have told you about protein. You should also know the tests for carbohydrates, for your aldehydes, your ketones, phenols, carboxylic acids, and all those tests. Right? They're actually very important.